Welcome everyone. Hope everybody is doing well. And I know it's almost finals week and it's kind of overwhelming. Hang in there, it's almost done. All right, let's start with that. Sorry, give me 1 second. All right. Before I start, I feel like I'm speaking to myself. So just interrupt me, ask me questions, or just interrupt me to just say anything. I'll be happy for that diversion there. All right. So today we'll slightly talk. All right. The agenda for today is more less details and more high level. So the first 30 or so minutes, I'll just talk through some of the third part of the workshop that we planned, which is to do with like data handling or analysis and data exploration kind of stuff. And then after that, we'll go into breakout rooms and we'll kind of plan what we'll do for hackathon. As you know, we moved hackathon to end of May, right? May 22nd, is that right? So we'll talk more about what the structure there is. We kind of scaled back some of the things we planned because our main objective here is for you all to learn, not feel overwhelmed. Enjoy the learning process and not feel like this is one more chore or homework that you have to do. All right, with that, okay, so the last two workshops, how many of you here attended the last two workshops? I guess the other way, let me ask you the other way. How many of you here did not attend either of the last two workshops? Just raise your hand or, I see, okay. So do you know where to find them? The recordings and the workshops, do you know where to find them? Can somebody unmute and just tell everyone where to find them?
Yeah, they're on the website, the EE website.
Great. Do you mind posting it on the chat so everybody knows where it is? Sure. Sorry, who spoke up just now?
Yeah, it was me, Will. I can do it.
Hey, thanks, Will. All right, great. Yeah, so by design, the last two workshops had a lot of information, as you may have realized. Very detailed, very low level information. That's partly because we don't expect you to know all of it, but we want you to treat it as a good resource moving forward, either for machine learning basics or some of the concepts introduced in the last two workshops will be super helpful for interviews as well. So make sure you use those resources well. And one other objective there is to ask your mentors or ask any of us questions, say if you're stuck somewhere or if you want to figure out how to run something. Okay, with that housekeeping out of the way, let's go into the overall summary. So we started off with this kind of machine learning abstraction, right? So we had three components. Can somebody tell me like what those three components were? Like any machine learning model, very high level components. All right. So we had input, we had a machine learning system, and we had an output. For now, let's just treat that system as a black box. It'll learn all possible combinations between inputs and outputs, and it'll figure out how to predict a certain output. On the input part, this was the focus of the first workshop, right? So we were talking about what are the different kinds of inputs. Can somebody tell me what different kinds of inputs we talked about? Anybody? I just get bored of listening to my own voice. So I would.
In workshop one.
Yeah.
I feel like people would know.
Okay. Well, I'll start asking the mentors then. Can somebody tell me what kind of inputs we talked about? All right, we talked about images or audio, right? There are different kinds of inputs. We can deal with either images or audio or video. So that workshop and the collab had details about how to read them, what different formats are there, and other details. Okay, And the big takeaway from that workshop was this concept of features and how to represent them, right? By the way, if somebody wants to add something to this, or if you think I'm misrepresenting something, speak up, please. Don't be shy. Okay. So the main takeaway from that was we deal with inputs. We have a concept of features. We learn how to represent these inputs to the system. For example, for images, we deal with unsigned integers between 0 and 255, right? Color images, let's say. Okay. Next, we talked about what are the different kinds of labels, right? So can somebody tell me what you do when we don't have labels for data? What kind of learning that is called? I mean, this is an interview question, by the way, if that may wake you up. Chris.
Sorry, could you repeat that, please?
Yeah. So what is the machine learning, I guess, branch called when we don't have labels for data?
Unlabeled data.
That's the data. What is the learning mechanism?
Okay.
Unsupervised learning, right? Because you have nothing to supervise it with. So that's where the output comes in, right? And when we talk about labels, we should also answer the question, what kind of labels? Are we talking numeric labels? Are we talking cats versus dogs? Or are we talking, I don't know, the weight of a cat? The weight of a cat would be a continuous number, right? So that would be a regression problem. So, okay. And we also briefly, Rimita and Dylan also briefly touched upon how do we measure the performance in the output space, right? Remember the whole confusion about the confusion matrix? Anybody? Okay, good. Okay. And finally, the fun part is the system itself, whether to classify or to regress. And the last two workshops, I hope we covered some idea of, you should be able to answer like questions like, say I have a cat versus dog, right? That's a problem. Would you classify or would you regress?
Classify.
Good, very good. Okay, and what about if you have cat, dog, and a turtle?
Classify.
Yeah, great. Okay, and the keywords were the classification or regression system, and we also talked about what kind of loss functions to choose, right? I'm not going to go into the details, but just key terms so you can go back and like look them up or Google them. And then the big part was model choice. What kind of models will you use, right? Whether you would choose a support vector machine, which is relatively simple model, or the fad these days, which is deep learning. Just forget about everything else. Most of the things deep learning will work. But no, that's the more cynical way of looking at it. It's important to kind of go back and go through some of the earlier machine learning systems so we can develop an intuition on how to use deep learning. That's the high level. Okay. So I hope most of you kind of got these three big topics or ideas from the last workshop, right? How to ask, how to operationalize a problem, a learning problem, and then be able to reason whether machine learning can solve the problem, and then how to solve the problem. Roughly, can I see some yes or no or some head nods? Good, thank you. All right. Okay, now here's the fun part. Anybody can build ML models. All you have to do is throw in your data, churn until you get some output. It's a big crank that you keep churning until you get an output. Then why can anybody build ML models? Partly because we have access to rich open source data sets, right? And we have access to resources and software, and there's a big push to make these open source. And finally, the internet. You can practically look up anything, and with zero computer science knowledge, well, maybe not, with almost zero computer science knowledge, you can build a machine learning model. But then what makes for a good machine learning design, right? Or what makes for a good machine learning model? There is a synergy between two different concepts. the modeling aspect of it and the analysis aspect of it. So the last two workshops, hopefully, you got some sense of what kind of modeling we can do when we have a certain problem. Today, we'll be kind of going into some details of what kind of analysis should we be looking at, even sometimes before starting modeling. So these are almost like two schools of thought when you have a controlled experiment, right? You can take all your variables and do some analysis and set up some tests to come up with some findings, or just put them into a machine learning model and classify and see how well you can classify it. That's a way of saying whether you can learn from the data. All right. So who makes these decisions? Can somebody tell me like who makes these decisions about the input, the output, and the system? Anybody? It's a trick question.
Yourself.
Yeah, exactly. Humans, right? We do that. We make these decisions. We make these decisions. What my input is, how my output label space should look like. Maybe I don't want to do cats versus dogs. I want to do pets versus humans, right? All these are decisions we make. So the first question you have to ask, whether you attend an ML talk or you're reading a paper, is sample. You want to be able to understand your sample. What is my sample? And then how to describe my sample. And we'll talk about bias for a bit. But for now, let's just focus on how to describe a sample. I'm going to go through a quick exercise, hopefully not too boring, in the Colab. So can you all see my screen? Okay, good. There's a lag. Give me 1 second. All right. Okay. So does anybody remember the iris data set that we kind of discussed last time? Yeah, okay, I see some nods, good. So if you look up the iris data set, you see these very high level summary, right? Are there more than one variable? How many samples do you have, and so on. So now let's try to see if we can describe the sample. When I say describe the sample, let's see what that means. First thing I would like all of you to kind of look up, spend 5 minutes, is this amazing Python library called Pandas. I can't tell you how many times that library has helped me throughout my research. Having that in your toolkit is a really important thing. Okay. So apparently this data set has these four different variables: the sepal length, the sepal width, the petal length, and the petal width. And you have some targets labels. So the task here was to classify one of three kinds of species, the iris flower species, whether it's a serosa, we'll see, we'll describe it. Great, so the next step is let's build a model, right? But do we care about what are petals and sepals? Or can we just take those numbers and plug them into a machine learning model? Anybody? Should we care? It's not a trick question. Yeah, we should care.
Yeah, we should
So,
care about
yeah,
them.
exactly. And this is something, this is what sets apart good machine learning designers or good computer scientists versus mediocre computer scientists. We do have to take a step back and look at what the hell we're talking about. What are petals and what are sepals? So you don't have to understand the biological functions, well, the biological details of it, but just to get a sense of what are we looking at in the physical world. Also, understand what these numbers mean. Like, what does 3.5 mean? In this case, it happens that it's a centimeter, it's a measurement. So they're measuring petals and sepals for 50 randomly drawn sample of each of these species. Great. Now let's describe a sample. Notice that I'm completely skipping over the pandas details. ping me if you're interested in it. I'm happy to run you through some examples. But the whole point of pandas is once you set up this kind of CSV kind of structure, it's very easy to now describe the data. You just say df, the data frame, that's a term pandas uses, and describe, and then it describes for you. So you can see, essentially, when we say sample description, that just means we want to be able to say how many samples we have in the data, right? And if it's a continuous measure, what is a mean, the standard deviation, minimum, maximum values. Can you see what I'm highlighting? Yeah, good. And the percentiles. So just as a refresher, actually, how many of you have done this in high school or college? Like, given a set of numbers, calculate the 25th percentile, calculate the median. You've done that, good, good. So you know what I'm talking about. And why do we care about this? I'll show you in a minute, but this is important to describe how these numbers, these variables are distributed. Great. All right, I'll stop there and I'll go back to what we were talking about. Right. So very briefly, we were able to answer what is my sample, how to describe my sample. You'll see that I'm using my or your because, as a ML designer, you own the data set, you own the design choices, so it's yours. So ownership is the first step. All right. Now, this is a really big part of any data set sample description, keeping an eye out for bias. How many of you have heard the term bias used in machine learning or good? So I understand this is not exactly a human-centered data set, right? Because we are talking about species and flowers, but can someone tell me what potential bias can creep in here? The data set we just looked at. Actually, let me ask you something. How many samples were in the data set we just looked at?
Is it 100 something? I think.
Yeah, 150. Good. So let's just go with 150. And what kind of potential bias can creep up with the sample we just collected? You can come up with crazy ideas.
Is it possible that out of the 150, we only captured a certain range of petal length, and there can be a potentially flowers of the same species with wider or longer petals?
Right. Yes. So you're talking about whether the sample was big enough. That's always a problem, right? What other kind of biases can creep in? Let's talk about, say we are just focusing on predicting these three kinds of species and nothing else, right? Now we want to deploy it, sell it to, what do you call horticulturists? Is that what you call them? For example, okay, let's go with that. So you want to say you want to sell this model to a horticulturist. What potential bias can creep in? What about, do you think it would be a problem that you collected all these flower measurements in England? That's what the data set initially did. And now you want to deploy this in US. Do you think that would make a difference? Right. So these are the kind of biases you want to keep an eye out for. In case of flowers, it's so much easier, but the complexity keeps going up as we are talking about more perceptual kind of things, right? Okay. With that, now we just talked about decisions. Let's take a small, let's go to the next step and talk about experimental design. So far, we only work with data sets that were given to us. So we didn't have to worry about how these were collected or how many samples were collected. We just assumed that it was a good representation of the problem we had, and we went with it. Right. Now, whenever you're setting up your own study, you will hear this term called experimental design quite a bit. So whenever you attend a talk or whenever you read a paper, keep an eye out for trying to identify these three components of any experimental design. If these three components are not clear, you can speak up. Like in a talk, for example, you can always ask, what is your independent variable? So hypothesis, right? That's let's put this aside for now. We'll come back to it. But in the Iris case, a hypothesis was predict a species for me. Okay. And then you have two kind of variables: the independent variable, the things that you can precisely sample or manipulate, right? If it's a control study, it's more about whether you're able to manipulate something. Like say you want to see how quickly a plant will wilt in different temperatures. So you can control the temperature, see if the plant will wilt. So that is an independent variable. In our case, it's about sampling. In the Iris case, we look at 50 flowers of the same species and we take the measurements. So we can say we can precisely sample whatever in centimeters. The dependent variable is something we should be able to accurately measure. Most of the problems come from this word accurately. So the dependent variable in our case was, what was the dependent variable in Iris case? The species type, right? The species type, in this case, we use an expert, say a botanist, to measure it, 0, 1, or 2. Good. All right, let's do the small exercise. I thought instead of me going on about this, we'll do like a small exercise and see if we can come up with a hypothetical study and see if we can set up some hypothesis. Ideally, I want like some crazy model, not cats versus dogs. That's too simple. That's too boring. So can somebody tell me just tell me, look around, whatever you're looking at, and say you want to come up with a hypothesis for something. So can somebody tell me what you want to test? I mean, I'm sure you thought about like being able to test something around your house. I know test is a loaded word, but. Or pick something from your class project. Sorry, go ahead, Chris.
Oh. Could we possibly test whether length in living in isolation affects like serotonin in terms of like how you feel emotionally or something? I think that's relevant to COVID.
No, that's a very valid question. Say it again. Try to make it, so most hypotheses are like a statement, right? Try to rephrase that. So you said isolation and emotion, right?
So the law.
Go ahead.
Could I just simply say like, the longer somebody lives by themselves, the more sad they will be?
Sorry, I have to subject you to my typing, but bear with me. Live in isolation. Does that represent what you said correctly?
Yeah. Sorry, I probably said it all messed up.
My internet is acting up. Okay, good. Now, what kind of features would we collect? Sorry, dependent variables or features, right? In our case, we were looking at, can you still hear me?
Yeah, we can hear you.
Okay, good. So in the flowers case, we were looking at some measurements, right? So what kind of features would you collect?
Maybe length and isolation.
Good. Okay, what else?
We could possibly, I don't know if this is realistic, but maybe collect some
Oh, the
data
whole point of
on-
this exercise is go as far as away from reality as possible, because isolation is too real for us. So let's make it hypothetical.
Like hormone data, like levels of serotonin,
Right.
or endorphins.
Let's just say serotonin. That's
Okay.
one hormone. Okay. What else? Come on, you must have all have. I definitely have so many features I can think of that crops up during isolation. Are
Potentially
you all
like
too young to drink alcohol? Probably. Okay, never mind. Sorry, go ahead.
time spent like on social media or
Good.
like
Yep.
communicating with somebody over social media or something. I don't know.
Yep, that's great. Two more and I'll let you go.
Maybe number of binge TV shows.
Absolutely. I think I may have the highest there.
Would like hours spent in bed also be one?
Absolutely. Excellent. Okay, good. Now, what is our independent variable here? This is the fun part. So you want to look at your hypothesis and you have to decide what your independent variable is, or rather what you want to accurately measure. What is it?
Sadness.
Yep, absolutely. Okay, how will you measure sadness? You ask people, right? You ask them if they're sad or not. So there's a lot more details there because now you're getting into are people capable of correctly answering their emotional state and so on. But this is the overall picture. So whenever you're thinking about setting up an experimental design, you often start with the hypothesis. You figure out all the features you can collect, which we call dependent variables, and then you go measure an independent variable and see what the correlation. See the predictive value of all these dependent variables for the independent variable. This is often how a well-designed study is done, but not always do we start with the hypothesis because we have a lot of data. We collect a lot of data sometimes in the wild. So we want to also sometimes go back and create some hypotheses by looking at the data. That's okay too. Just closing the loop there. Okay. Now, what we talked about what makes a good ML design, right? We talked about how to collect your sample, how to think about experimental design, and keeping an eye out for biases. Now, what makes for an expert ML design, which you are all going to be at some point, knowing that you're all in computer science. So what makes for an expert ML design? This I cannot shout loud enough. It's all about looking at the down data. Most ML people don't do is what they don't do is looking at the data. Most people just take the data, plug it into a machine and start churning. Not okay. You have to look at your data. Of course, there are other important things too, like you have to understand where you're deploying, right? You can't take your flowers model that you built for England and directly deploy it to US without thinking about how well it performs, for example. And also understanding the why, right? Why did you measure petals and sepals only? Why not other stuff? Maybe color, right? Okay, but I'm going on about looking at the damn data. So how do you look at the data? So let me go through a quick sample exploration. This is really high level exploration, but let's take a look at it. Okay, another library here, something you may want to note and look up when you have some time is a Python visualization library called Seaborn. Seaborn as in like so. I've highlighted it here. Okay. Look it up. It's amazing. I've used it so many times. All right. So I'm just going to highlight 3 different things here. looking at data distributions, feature correlations, and statistical testing. I'm not going to go into statistical testing details, but just so you're aware that it exists. Okay, so let's start with distributions. So I looked at petal length. So we know what petal lengths are, right? We looked at the images, we got a sense of what petal lengths are. And we can plot the distribution. So you see the petal length on the x-axis is centimeters. So it ranges from what, some less than a centimeter to over 7 centimeters, right? And you can see what the counts are. Now, immediately with distributions, you can see that this is not a uniform, sorry, not a, anyway, it has two modes. There's one peak here and there's another peak here. So it's called a bimodal distribution. You don't have to worry about that, but you can get that from this figure.
Krishna, do you want to just briefly explain what the what a histogram means?
Yes. So the histogram is a way of representing data by binning it. So say you have, let's see, we described the sample just now, right? So we said the petal length here, the petal length goes from 1 centimeter to 6.9 centimeters. So you go through every centimeter, say you go anything less than 1 centimeter, and you count how many of them, how many samples you had in that bin. That's a term they use. And then you look at that same count between 1 and 2. That's a good question, Amrita, thanks. Yeah, and that's how you get this distribution plot. It's a histogram plot, but it'll tell you a lot about the data. You should be able to look into it and say there are two modes, and there is no, in fact, no samples between this. So ideally, if you want to collect a good sample, you want to be able to collect a wide range of petal lengths, right? Now, the fun part is you can directly overlay the label, that is the species type, onto this distribution and see what's going on, like so. So I've overlaid the labels for the 3 species. So clearly, you can come up with a very simple decision rule to classify setosa versus rest. Can somebody tell me what that rule would be by looking at this plot?
They're categorized based on their petal length.
Yeah, and what would that rule be? Sorry, so say I want you to build a very simple rule-based system, which would tell me that just by looking at this plot, which would tell me that some species is setosa or not. How would you do that?
Can you think of it as like an if-else condition?
Yeah.
Like if something, it would be setosa. If something else.
Oh, if I don't know what the measurement for. What are the units down there?
Centimeters.
Or
good. Yeah.
Like if the petal length is between 0 to 2 centimeters, it would be setosa, and then 3 through 5, and then 5 through 7 or 8.
Right. Obviously, this is a very simple toy data, and we are not usually so blessed with other data sets we work in real life. But this should give you an overall sense of what's going on. Okay, now let's switch gears and talk about correlation. So most of you are familiar with correlation, right? Okay, so you know what a magnitude and sign means. So what a negative correlation, what a positive correlation means. Good, so I don't have to go into that. All right, so let's look into our 4 features we were interested in: the sepal length and the width, and the petal length and the width. And again, I'm using Seaborn to plot this, but I've plotted the correlation. Can somebody tell me if this matrix is symmetric? Yes,
it
no?
does appear to be symmetric, yeah.
Great, yeah. So most correlation matrices are symmetric because you look at correlation between length, sepal length versus sepal width, or you switch the x and y, you'd get the same number. So just so we get that out of the way. Okay, now what does this number mean? So you should be able to kind of a big part of data exploration is to be able to interpret the numbers, not just plot the numbers. So we should be able to say things like, in the sample we have, petal length and sepal length are highly correlated because you see a 0.87 positively highly correlated. And you can also look at petal length and sepal width and say it's negatively correlated, not highly, moderately. Again, these are rules of thumb. Rules of thumb. Things like anything below 0.5 is moderate, anything over 0.5 is high. Okay. Oh, what can somebody tell me why this is 1? Why is petal width highly correlated with petal width? The same variable, right? So that's the maximum correlation you can get. And the minimum correlation you can get is -1, the other way. Okay. But what is exactly correlation capturing? It's capturing this kind of pattern. So this is often called a scatter plot, which you're all, I believe, familiar with. So it's capturing this linear relationship. You see, as I increase the petal length, say I can manipulate all these 2 variables, right? Petal length and petal width. It happens that as I increase the petal length, the petal width also increases. That's what this physically means. Now I can also overlay, right? Now I can also overlay labels on this, and you can now come up with a slightly complicated decision rule, right? Petal length, not only should the petal length be less than 2 centimeters, but the petal width should also be less than 1 centimeter. Only then it is setosub. So you're slightly complicating your model. That's the overall idea. Okay. I'm going to skip this, but I'm happy to talk through all of this. This is another case of sepal length versus sepal width, where you don't see a lot of correlation, right? You can see, like in this case, you don't really see a lot of correlation. It's hard to draw like a line with a positive slope. In fact, you can slightly draw a line with slight negative slope, probably. And that's true. Like, let's look at sepal length and sepal width. Where is it? Yeah, you see here, it's -0.12. So there's slight negative slope there. However, if you look at specific species, like the setosa here, there's a positive correlation. You can draw a positive line with a positive slope. That's the overall idea. All right, now you can completely blow this up and look at all possible combinations of correlation. I've always find this useful in Seaborn, this command called pair plot. You don't need to remember, just something to know that it exists. You can plot something like this. Let me see if I can get
What
that
is
all on the
it?
screen.
What is it that you're using to plot? What does the SN stand for?
Seaborn.
Okay.
Yeah, good. Yeah, so now you can plot all combinations of features like this. Not only do I see the distribution, right? This is a smooth distribution. Let's just go with that. But also you can see how their values are distributed. So in this case, let's see. These two, let's look at these two, petal width and sepal length. So this is how they're correlated. Clearly there's a very high positive correlation, right? And petal width and sepal length, that the distribution would be that, right? Is that right? No, yeah. No, just the sepal length distribution would be this. So you can see there's a clear separation between the different species, but not so much in sepal width. Probably petal width is the most useful, right? Because all 3 species are separated. So this is why we care about looking at the data. So we want to look at the data and develop some intuition about what's going on. This is a toy data set. It becomes easier to look at this. But as and when you look at more complex data sets, you have to find creative ways of looking at the data. I think it starts off, yeah.
No, I just wanted to say, like, so one part of machine learning is also trying to find out what are like what we call discriminative features.
Yep.
Which means, like, see, out of these like 4 different features right now, like there are
Yep.
2 features that can like distinguish different classes. Like all the classes are shown in like blue, orange, and green here. So like you can just look at this plot and be like petal length and petal width can really discriminate between the different classes, whereas
Right.
like sepal width cannot.
may not be very
because
helpful, right?
it might not. Yeah.
Right. And all the while, remember, there's a physical meaning to these things, right? So we can always go back and look at, okay, does this make sense? We can always go back to what we started with and kind of try to reason if that makes sense. So that kind of closes the loop. Not only do we look at the data, we should be able to interpret it within reason. Of course, you can't do this when you're working with high dimension data. All right. I'm just gonna mention a couple of terms in statistical testing. I'm not gonna go into the details, but this is the analysis part of it. Say now, Amrita was mentioning discriminative features, right? So we want to say, we want to quote unquote test if this sepal length is quote unquote significantly different between setosa and virginica. How do you do that? That is called a hypothesis testing. So you set up a hypothesis, you use a statistical test for under many conditions, and then you check if they are significantly different or not. I'll just leave it at that. But if somebody wants to talk to me about statistics, I'm more than happy to talk to you about that. All right. That's the end of today's talk. Any questions, comments, anything at all? Does it give you an overall sense of like what experimental design is? If somebody asks you to run a study, you can kind of reason about it within if it's a controlled environment, right? Okay. Now we did all this because I know many of you have been thinking about projects for the hackathon, right? Hey, Rachel, what's up?
Hi, I just had a really quick question.
Yeah, please.
How does machine learning kind of interact with categorical data that's like not numbers?
Good. There's something called one-hot encoding. Are you kind of familiar with it? Okay, so say we are looking at cats and dogs, right? They are categorical variables. So one way to represent a cat is a 0, 1, and a dog is a 1, 0. That will kind of take care of it. Actually, that's probably an overkill. 0 and 1 will do the job in this case, right? So that's one way to represent something. So let's go back to this, right? So the 3 questions you want to ask is, say we are dealing with categorical inputs. So how do we represent them? One way is a one-hot encoding. Right? The other way is in the label space, obviously, you classify. So 0 and 1 will do the job. Kind of sort of answer the question? Okay.
I've just like kind of heard machine learning in reference to like you'll train like a machine to say like write a script, like you'll feed it like a lot of like things.
Yes.
How does that kind of work?
Yeah, good question. It's a similar idea, but a little more involved. So for that, what do you need for a set of words, right? So you start by building a vocabulary. So different vocabulary, very simplistically, you can think of, now you can take the vocabulary of say, I don't know, 50,000 words and still do a one-hot encoding for them. It'll be a 50,000 long vector, but you can still do it. And then you introduce other tokens, commas, periods, exclamation points, all of that. And then you train a network to be able to predict it. I'm of course brushing in a lot of details, but that's the overall idea.
Okay, got it. Thank you.
Those are also like generative models that are very different from the kind of models that we talked about here. So here we are talking about like discriminative models, like models
Right.
that can like differentiate between different classes. But like something like predict, like writing a paragraph would be like a generative model. So
Right.
it operates differently, but like kind of like what Krishna said. But the way you train and everything's like slightly different.
Actually, that's a good point to make. Thanks, Amrita, about generative models, right? A simple example is, say I'll ask you to create a new setosa flower. So one way to do that is you sample from the petal length, you sample from the petal width, sepal length, sepal width, and put it together and create your own flower. That would be a generative model. What we're doing right now is just tell me if this is setosa or not, right? That's the general idea. Good question. Thank you. Any other questions? All right. We have like 40 minutes. I'm gonna go, oh, I was talking about projects. So the idea is we'll break out into 20-minute sessions, 20-minute breakout sessions, and like discuss what next steps. But before that, let's talk about hackathon. How many of you are anxious about the project and the hackathon? Okay, first thing, don't be anxious. The exercise here is not to, it's not a homework. The exercise here is come up with a problem, think through it. That's all we're asking you. We try to kind of create a structure that may help you to kind of answer some of the questions for the hackathon. So I'm going to quickly go through it, but hopefully you get to ask more questions to the mentors and get some insights too. Okay, obviously, like most projects, you start off with a project title, right? And then you want to ask the question in your projects, like, what's the topic? What are we doing? Keep this very high level. It's a moonshot idea, like the example we work with, right? You want to see if how is isolation correlated with sadness? That's the high level idea. And you want to also be able to talk about the impact. How does it impact people and society? And the goal, the objective topic is not to be specific about, I'm going to predict if I'm sad using these 5 features. No, it's about, I want to predict if I'm sad and how it relates to isolation. Right. Okay. Next, we talk about motivation. Why are we doing this? Obviously, because we are all feeling isolated. And then we talk about considerations. We kind of went through some of these, right? What are the features we want to think about? And then details. How many people are you going to ask these questions? How are you going to measure serotonin? How are you going to ask them if they're sad? When are you going to ask them if they're sad? All these are considerations. And how are you going to implement it? You can build a simple decision-based model, just like if else we did, right? Just plot out the distributions and come up with a rule. That's a simple implementation you can get. And we want you to ideally come up with one big finding. Like say something like, in my data set, I found this X percent of the time. In a data set I collected from 10 people, I found that in 6 people, there was a high correlation between sadness and isolation. Isolation is measured by whatever variables, right? Again, this is more details than you may require, and your mentors will kind of fill you in about that this is not a rigid rule, right? You can either do a demo, you can just explore some space, think through it, and kind of come up with some hypotheses. The objective here is not to run a big model on a big data set. If you want to do that, we are happy to support you and encourage you. But that's why I'm saying don't get anxious about it. That's not the objective here. The objective here is to think through the experimental design we just talked about. If I had all the time and resources, this is how I would build it. And let me see if I can set up a small toy problem with the skills I learned in the past 3 workshops and show you something in my for the hackathon. That's sort of the high level idea. Sounds good? Bad? Any questions? Great. All right, I'm gonna stop sharing so I can look at you all. All right, so we have what, like 20 minutes? So can we go in? Do you have any questions before we go into a breakout session?